Hey, welcome back to Cellular Respiration. Last week we talked about anaerobic respiration when we started with glycolysis. Now we're going to take that pyruvate molecule that we made during glycolysis and we're going to transfer that into the citric acid cycle to continue our process of deriving some ATP or energy from our sugars that we're intaking as chemical energy. So our pyruvate molecules that we made are going to get transferred into the mitochondria because remember they were free floating in the cytoplasm where glycolysis takes place. They will then become acetyl-CoA, and we're going to talk about the process of how that becomes. And CoA itself is actually a derivative of vitamin B5. So in order for this process to occur, we need vitamin B5. So it's really important to just have all of your B vitamins, be taking these supplements so that you're able to make energy as efficiently as you can. So let's break down that pyruvate molecule. A carboxyl group is removed and released as CO2, or carbon dioxide, via the enzyme pyruvate dehydrogenase. Electrons are then picked up by the electron carrier NAD, which is then reduced to form NADH. And then the acetyl group combines with CoA to form acetyl-CoA. Acetyl-CoA will then donate an acetyl group to oxaloacetate, which is one of our first products in the citric acid cycle, to form citrate. And then that is the start of the citric acid cycle. You may have also heard it be called the Krebs cycle or the TCA cycle. Any of the above will work. When acetyl-CoA donates that acetyl group to oxaloacetate, the CoA then goes back to being recycled to form more acetyl-CoAs from the pyruvates that are coming in. So the citric acid cycle is going to harvest any of the remaining energy from that original glucose molecule that was formed into this pyruvate molecule. And then it's going to take that energy and shuttle it over to the electron transport chain to see how much ATP we can harvest out of that. So this is an overview of the cycle. I know it looks like a lot, but if you ignore the actual chains of carbons here, the structures, it doesn't look quite so bad. So we're going to go step by step. So the citric acid cycle takes place in the mitochondrial matrix. After we're all said and done here, we will have formed two CO2 molecules, one GTP or ATP, which will depend on where we're at in our tissues, one NAD and one FADH2. Both NADH and FADH2 will donate electrons to the electron transport chain for this process to continue. The citric acid cycle does not directly consume oxygen, and really it makes very little ATP by itself. For such a complex process, we're only going to get out of this with either one GTP or one ATP. It's kind of sad. It's not a very efficient process. It acts as an intermediate between this glycolysis and the electron transport chain. So in our first step, we've already taken pyruvate, sometimes called pyruvic acid, and turned it into acetyl-CoA. Well, that acetyl group and oxaloacetate together form citrate. That is an irreversible step. It's highly exergonic, and it's controlled by negative feedback. So if we have a lot of ATP in our system, this step won't happen. And that's our body's way of not producing too much energy that we can't use. In our second step, citrate will become isocitrate, which is its isomer, which is why it's called isocitrate. In the third step, isocitrate will be oxidized to form alpha-ketoglutarate. It will also form CO2 and 2 electrons via the reduction of NAD into NADH as byproducts. This step is also regulated by negative feedback of ATP and NADH. So if there's too much ATP or NADH, this step won't happen. If there's a lot of ADP, this process will be kind of catalyzed through. It'll run a little bit faster. In step 4, alpha-ketoglutarate will become succinyl-CoA. This step also releases electrons by the reduction of NAD into NADH. And this is also regulated by feedback inhibition of ATP, succinyl-CoA, and NADH. If there's too much of any of those substances, this step won't happen. In step 5, a phosphate group is substituted for CoA, forming a high-energy bond. So succinyl-CoA will become succinate, and this will form either GTP or ATP. It depends on the tissue. So if we're in a tissue that uses a lot of anabolic reactions, we're making a lot of products, like in the liver, then we're going to want to form GTP. In other areas of the body where we use a lot of energy, things like muscles, your brain, your nervous tissue in general, you're going to want to form ATP. In step 6, that succinate was dehydrated to form fumarate. FAD becomes reduced into FADH2, and those electrons that are transferred there go straight to the electron transport chain, which we will talk about in just a few moments. In step 7, fumarate undergoes hydrolysis to become malate, and then malate becomes oxidized to form oxaloacetate, our beginning product. Another molecule of NADH is produced here. So overall, we made 3 NADH molecules, 1 FADH2, and 1 GTP or ATP. Now this cycle is considered amphibolic because it both breaks things down and makes things. So it's both catabolic and anabolic. Its intermediate compounds that are made are used to synthesize amino acids. So that was all the Krebs cycle. Now we're going into the electron transport chain. This is also known as oxidative phosphorylation. This generates the most ATP out of all of your cellular respiration processes. We do this by accumulating hydrogen atoms, and hydrogen atoms are going to be a carrier of energy that we're going to use and harness to create ATP. So hydrogen atoms will accumulate in this intermembranous space. They will diffuse into the mitochondrial matrix via ATP synthase, which is an enzyme. And this phosphorylates ADP into ATP. You didn't think this was going to be uncomplicated, right? This is very complicated. So the electron transport chain, it's our last stop in the formation of ATP. It's the last component of aerobic respiration, and it's the only component that actually uses atmospheric oxygen, the oxygen that you're breathing in. This is a series of redox reactions, which is what we talked about last week in the first slide. Combination of oxidation and reduction. So, going back for a second, this is an overview of the entire electron transport chain. We have all of these complexes that are numbered in Roman numerals, so 1, 2, 3, and 4. We also have some cytochromes and a Q complex. So, just to orient you a little bit. They all sit as integral proteins within the intermitochondrial membrane. Some are peripheral proteins like cytochrome C. And they're going to be shuttling these hydrogen ions into the intermembrane space from the mitochondrial matrix. So let's start with complex 1. 2 electrons will be carried by NADH. NADH will be oxidized into NAD. This complex is composed of something called FMN, which is derived from vitamin B2, which is riboflavin, and an iron-sulfur containing protein that we just abbreviate Fe for iron and S for sulfur. The enzyme that catalyzes this process is called NADH dehydrogenase. We're taking NADH and turning it into NAD. In this process, we're pumping 4 hydrogen ions across the membrane into the intermembrane space. Next, we get to Q and complex 2. This complex is going to directly receive that FADH2 that we were talking about from the Krebs cycle. A protein called ubiquinone will connect complexes 1 and 2 with complex 3. Q is going to receive electrons from NADH from the first complex and the electrons from FADH2, so it's an electron carrier. But FADH2 is a less efficient electron carrier than NADH, so we will make fewer ATP molecules from FADH2. The number of ATP molecules that are obtained at the end of this process is directly proportional to the number of protons that are pumped across the inner mitochondrial membrane into the intermembranous space. Complex 3 is composed of some proteins cytochrome B and cytochrome C. The complex is known as a cytochrome oxidoreductase. We're going to transfer some electrons here and turn iron 2+ into iron 3+. Complex 3 will pump protons through the membrane, and it will also pass electrons on to cytochrome C for transport to complex 4. So you can see that each complex is transferring electrons to each other while pumping ions across the membrane. So the only transfer between complexes is of electrons. Ions are being pumped across the membrane. Cytochrome C will also receive electrons from Q. So from Q and from complex 3 are electrons that are going to cytochrome C, which is just a mediator to pass electrons on to complex 4. Complex 4 is comprised of proteins that are cytochrome proteins CA and A3. It has 2 heme groups, which is just an iron complex, and 3 copper ions. At this complex, we have a reduced oxygen, which will pick up 2 hydrogen ions that have been made to make water that will leave as a byproduct, which helps to form our gradient for chemiosmosis. So note that we have electrons being passed among all of these complexes, and you can see the electrons being passed via the arrows. Across the membrane, all that's going across the membrane are the hydrogen ions, which will be used to form ATP. So the free energy from those redox reactions is used to pump those hydrogen ions across that membrane to begin with. We have this enzyme ATP synthase that will act as a generator. So it takes those hydrogen ions and converts them into ATP. So hydrogen ions flow through it. It uses that potential energy of the hydrogen ions to convert ADP to ATP, phosphorylates it. So this process generates 90% of the ATP that we use. Our final electron acceptor will be oxygen. The number of ions that pump across that mitochondrial membrane depends on the species. We extract about 34% of the energy from glucose, and the rest is released as heat energy, which, as we know, is unusable. So for all of the effort that it took for us to make that energy, it's a pretty inefficient process. So if we're not in the presence of oxygen, if aerobic respiration does not occur, we will have anaerobic cellular respiration, which comes in the form of fermentation. So in this case, NADH will be oxidized into NAD. And it does so by using an organic molecule to regenerate this process, to regenerate this electron carrier. Anaerobic cellular respiration occurs in some prokaryotes. For example, a specific type of bacteria called methanogens reduce carbon dioxide, which would be an inorganic molecule, into methane to oxidize NADH to form NAD+. These methanogens are found in soil and also the gastrointestinal tract of some ruminants like cows. Lactic acid fermentation also is used by animals and certain bacteria, also red blood cells and skeletal muscles. Lactate is then sent on to be metabolized by the liver by the enzyme lactate dehydrogenase. People used to think that when you work out a lot and you get sore, it's because you have this buildup of lactic acid in your muscles, but that hypothesis has been disproven. So it's more so likely due to the fact that your muscles are kind of breaking down and degenerating because you didn't take enough protein in after you beat them up a lot. Alcohol fermentation produces ethanol from that pyruvic acid that we made through glycolysis, or pyruvate. Other types of fermentation. So some bacteria are anaerobic, or they can be anaerobic. So if they're facultatively anaerobic, that means that they can transition from anaerobic to aerobic, meaning they can live in either oxygen environments or oxygen-free environments. Some bacteria are obligate anaerobes, where they will die in the presence of oxygen. The species that are obligate anaerobes, one really good example is Clostridia, which causes things like tetanus, gangrene, C. diff. Those are all obligate anaerobes. So when they get into your skin, they can cause this fermentation-like process and break tissues down at a high rate of speed. Glycogen is the storage form of sugar, and it is stored in both liver and muscles. When your body is at a state of low blood sugar, it will be broken down by the hormone glucagon. This allows ATP to be produced for a longer period of time during exercise, which is why you often feel more energized after you exercise. Catabolism of fructose and sucrose produce about the same amount of ATP as glucose. Amino acids can also be recycled into the synthesis of new proteins, and they can also be shunted into the glucose catabolism pathway when your body is in ketosis, which is a state of like pseudo-starvation. The breakdown results in urea as a waste product, and this is kind of what the body does during a ketogenic diet. You're in a state of ketosis and using these amino acids as fuel. Lipids such as cholesterol and triglycerides can be made and broken down via this glucose metabolism. They can be catabolized via beta oxidation into acetyl groups to form acetyl-CoA, which can contribute to the TCA cycle. Cellular respiration has to be regulated to produce a balanced amount of ATP. It's already not an efficient process, so if you're making more than you need to use, then your body is just making all of this energy and using energy to create this energy that you don't need. So if energy is not being used to the extent that it should be, that can be shunted to appropriate places to make other things that we need, like our macros, proteins, carbs, and nucleic acids. There are some other regulatory mechanisms in place. We have GLUT proteins that are in specific types of tissues, which kind of turn on and off depending on what our insulin is telling our bodies. If insulin is active, then it's telling these tissues to allow glucose to enter, and essentially get out of the bloodstream. Allosteric effectors can either increase or decrease the enzyme based on the conditions. Remember our allosteric inhibitors and activators. Some examples of allosteric effectors in this case are ATP, ADH, AMP, NAD+, and NADH. So remember, glycolysis begins with hexokinase, the enzyme, which phosphorylates glucose so that it can no longer leave the cell membrane. So when this enzyme is inhibited, glucose can diffuse out of the cell. That's a way to regulate glycolysis. Remember, PFK or phosphofructokinase is the main enzyme that is controlled in glycolysis. It's our rate-limiting enzyme. Its activity is decreased by an increased level of ATP, citrate, and even an acidic pH. Pyruvate kinase, which creates pyruvate, is also regulated by too much ATP and phosphatase. Having acetyl groups or too high of NADH means that less pyruvate can be converted into acetyl-CoA. In the citric acid cycle, our rate-limiting enzymes are alpha-ketoglutarate dehydrogenase and isocitrate dehydrogenase. The citric acid cycle itself is also inhibited by ATP, NADH, and succinyl-CoA. And the electron transport chain is affected by ADP and ATP. As you can imagine, if you have a lot of ADP, the electron transport chain would speed up, and if you have too much ATP, it would slow down.